This is Sarah May Ligasa from Bachelor of Secondary Education, major in Science. And for today's video, I will be very glad to share with you my insights and learnings from Biology 2 Genetics, specifically from Unit 1 to 3. In the mid-80s, the revolutionary work of Charles Darwin and Gregor Mendel set the stage for the rapid development of genetics in the 20th and 21st centuries. Greatly influenced by his voyage on the HMS Beagle, Darwin's thinking led him to formulate the theory of natural selection, which presented an explanation of the mechanism of evolutionary change. Darwin, however, lacked an understanding of the genetic basis of variation and inheritance, a gap that left his theory open to reasonable criticism into the 20th century. Shortly after Darwin published his book, Gregor Johann Mendel published a paper in 1866 showing how traits were passed from generation to generation in pea plants and offering a general model of how traits are formed. There are three scopes of genetics. The first one is transmission genetics. Second, molecular and biochemical genetics. And lastly, population and biometrical genetics. Transmission genetics, it includes the basic principles of genetics. Transmission of genetic material from one generation to the other. Molecular and biochemical genetics is the study of the chemical nature or the structure and function of genes. Population and biometrical genetics is the study of the behavior and effects of genes in population, often using mathematical models. DNA technology is in the news almost every day. Most often, the topic is a new and promising application in medicine. But this is just one of numerous fields benefiting from DNA technology and genetic engineering. One important use of DNA technology is the identification of human genes whose mutation plays a role in genetic diseases. These discoveries may lead to ways of diagnosing, treating, and even preventing such conditions. The introduction of genes into an afflicted individual for therapeutic purposes holds great potential for treating the relatively small number of disorders traceable to a single defective gene. DNA technology enables scientists to produce transgenic animals which speed up the selective breeding process. The goals of creating a transgenic animal are often the same as the goals of traditional breeding. For instance, to make a sheep with better quality wool, a pig with leaner meat, or a cow that will mature in a shorter time. DNA testing, on the other hand, can identify the guilty individual with a high degree of certainty because the DNA sequence of every person is unique except for identical twins. The pharmaceutical industry derives significant benefit from advances in DNA technology and genetic research, applying them to the development of useful drugs to treat diseases. Babies who have deficiency could be treated with additions being done to their genetic structure. Those who cannot reproduce due to medical complications have also seen positive results with surrogate parents concept becoming available due to DNA technology. Increasingly, the remarkable ability of certain microorganisms to transform chemicals is being exploited for environmental cleanup. If the growth needs of such microbes make them unsuitable for direct use, scientists can now transfer the genes for their valuable metabolic capabilities into other microorganisms which can then be used to treat environmental problems. Biologists have traditionally classified all living organisms into two major groups, the prokaryotes and the eukaryotes. A prokaryote is a unicellular organism with a relatively simple cell structure. A eukaryote has a compartmentalized cell structure with components bounded by intracellular membranes. Eukaryotes may be either unicellular or multicellular. For any cell to reproduce successfully, three fundamental events must take place. Its genetic information must be copied. The copies must be separated from each other, and the cell must divide. All cellular reproduction includes these three events, but the processes that lead to these events differ in prokaryotic and eukaryotic cells because of their structural differences. When a prokaryotic cell reproduces, its circular chromosome replicates, and the cell divides in the process called binary fission. Replication usually begins at a specific place on the circular chromosome called the origin of replication. Like a prokaryotic cell reproduction, eukaryotic cell reproduction requires the processes of DNA replication, copy separation, and division of the cytoplasm. However, the presence of multiple DNA molecules requires a more complex mechanism to ensure that exactly one copy of each DNA molecule ends up in each of the new cells. The cell cycle takes place in cells that are actively dividing. A new cell begins after a cell has divided and produced two new cells. Each new cell grows, develops, and carries out the functions specific to its cell type. At the end of the cycle, the cell divides to produce two cells, which can then undergo additional cell cycles. The cell cycle consists of two major phases. The first is interphase, the period between cell divisions in which cell grows, develops, and functions. In interphase, critical events necessary for cell division also take place. The second major phase is the mitotic phase, the period of active cell division. The M phase includes mitosis, the process of nuclear division, and cytokinesis or cytoplasmic division. Meiosis is distinguished from mitosis by three events of meiosis 1. The prophase 1. Each homologous pair undergoes synapsis and crossing over between non-sister chromatids with the subsequent appearance of chiasmata. The metaphase 1. Chromosomes line up as homologous pairs in the metaphase plate. And lastly, the anaphase 1. Homologs separate from each other. Sister chromatids remain joined at the centromere. Mendel's law of segregation encapsulates this general principle of heredity. The two alleles for each trait separate or segregate during gamete formation, and then unite at random, one from each parent at fertilization. The term segregation refers to such equal segregation in which one allele and only one allele of each gene goes to each gamete. Punnett square is a handy diagrammatic device for predicting the allele composition of offspring from a cross between individuals of known genetic makeup. Notice that we use a capital letter to symbolize a dominant allele and a lowercase letter for a recessive allele. Breeding an organism of unknown genotype with a recessive homozygote is called a test cross because it can reveal the genotype of that organism. The test cross was devised by Mendel and continues to be an important tool used by geneticists. A test cross involves mating an unknown genotypic individual with a known homozygous recessive. Law of independent assortment, which states that two or more genes assort independently, each pair of alleles segregates independently during gamete formation. For some genes, however, neither allele is completely dominant, and the F1 hybrids have a phenotypic somewhere between those of the two parental varieties. This phenomenon, called incomplete dominance, is seen when red snapdragons are crossed with white snapdragons, and all the F1 hybrids have pink flowers. Another variation in dominance relationship between alleles is called codominance. In this variation, the two alleles each affect the phenotype in separate, distinguishable ways. So that's it, guys! Thank you so much for watching, and I hope you have learned something. Again, this is Sarah May Ligasa from Bachelor of Secondary Education, major in Science. Thank you!